Morning class. In this lecture, we'll be looking at the other approaches to identification of bacteria. One of the ways we can identify bacteria is by using serological methods. As microbes are antigenic, they are able to elicit immune response. Okay, so there are antiserum against specific bacteria that is available commercially that we can use to test against the isolates that you have to see whether they are the bacteria that you want. So this serological test allows rapid testing because this is usually done through the slight agglutination test. So what this slight agglutination test usually incurs is you'll be given a slide, usually dark in color. Then what you do is you place the antiserum, one drop of antiserum, and mix it with your culture, with your isolate. Okay. Then what you do is you swirl the slide. If the antiserum is positive against your isolate, it will agglutinate your bacteria. It will clump your bacteria, and you can see clumps on your slide. However, if it is negative, there is no agglutination, no clumping. What you see is just a smear, okay? A smear of bacteria in your antiserum. So that's the slight agglutination test. In bacteriology, it's useful for salmonella, the identification of salmonella, because there are more than 2,400 serovars. It's also important for Vibrio, especially in the determination, in the identification of the Vibrio cholera O1 strain. The O1 strain is the cholera strain, the strain that causes epidemic cholera. Okay, so Vibrio cholera O1 versus non-O1. So that's a very important step in the identification of Vibrio cholera. It is also useful for Streptococcus pneumonia. Okay. So when we do this serological test, it is usually done with positive and negative controls. And nowadays, they also use ELISA and Western blotting. When you have all this, let's just say a new serological test that you want to check to identify a new pathogen, it is important to know the sensitivity and specificity of the test. Basically, you have to have a sample of positive and negative controls, and then you use your test to on these samples, and you determine how many were positive correctly and false negative. Okay, and how many were correct negative and false positive. So these are the two terms that you have to familiarize yourself with: sensitivity and specificity. So sensitivity measures the frequency of correct positive results. Okay, so correct positive over correct positive and false negative. So the frequency of correct positive over all the infected samples. So whereas for specificity, it is the correct negative over all the negative samples. Okay. So For example, if between sensitivity and specificity, probably sensitivity is more important. For example, it is not feasible for a test to have false negatives when the person is infected. Okay, for in the case of COVID-19, the sensitivity of some of the rapid test kits is not 100. It's not even 100%. Okay, it's below 90%. So that means there are infected persons who can still be tested negative. So that is not good for the control of the infection. Okay, so sensitivity, it should be close to 100%. Okay? But the percentage also depends on how big your sample size is, because the bigger the sample size, of course, the better the more confident you are in your test. Other methods of identification for bacteria, these are mostly for you also have bacteriocin typing. So bacteriocin typing is very specific for lactobacillus. Okay, for bacteria that produces bacteriocin. Okay, bacteriocin is a small uh, metabolite that has antimicrobial properties. Other methods of identification can also use the molecular approach, for example, the G plus C ratio, the GC ratio, related to the melting point, and DNA fingerprinting, DNA sequencing, and also uh, nucleic acid hybridization approaches. So when we are using biochemical test methods, uh, there are two approaches that we can use. One is the dichotomous key method. The other is the blunderbuss approach. So for dichotomous, it's mostly for identification. It is a stepwise sequential system. Okay, and only determinative tests are adopted. Because it is stepwise and sequential, it takes time, because for every test, you probably have to wait for the bacteria to grow and to produce the results before you can continue to the next step. Then it has also it has lower predictivity, because a mistake in one of the steps will result in concurrent errors. Okay, so the probability for correct identification is low. This is an example of a dichotomous key, where, for example, if you have a bacterial isolate, you do a simple stain first to see what shape it is. Then you carry out a gram stain to see whether it's positive or negative. Sometimes you can immediately go for a gram stain, because after gram staining you can also still see the shape. Okay, so if it's a cocci, then if you have gram negative cocci, then probably it's a Neisseria. Whereas if it's a gram positive cocci, then you are left with the it's either probably Streptococcus or Staphylococcus. For bacilli, if it's gram positive, spore staining is important, acid fast staining is important. That will tell you whether it's a mycobacterium for acid fast, and if it's a endospore, it'll tell you whether you have bacillus, or if not, other bacteria. Okay, so even in the endospore staining, if it's bulging, then you know it's probably Clostridium. If it doesn't bulge, then it's most probably a bacillus. Okay, so but these are all presumptive identification only. You'll need a lot more tests to ensure a better identification. So if it's gram negative, you use McConkey. to see whether the bacteria utilizes ferments lactose. If it ferments lactose, it becomes pink. If not, there's no color here. So this is a very general dichotomous key for both gram-positive and gram-negative isolates. So if you see here, dichotomous key, you can make your own dichotomous key, okay? Because in every lab, it also depends on what are the bacteria that is more frequently uh, isolated, and also what are the tests available in your lab. So from there, you can draw out a dichotomous key suitable for use in your lab. Uh, so there is no single correct way to write a key. Okay. So the goal here is for presumptive identification of a bacteria. So this is a dichotomous key for gram-negative heterotrophic bacteria. So it's a shape. After gram staining, you divide them, separate them out according to shape. So if it's cocci, gram-negative cocci, most of the bacteria the presumptive identification should be Neisseria, the genus Neisseria. Whereas if it's rods, then you look for oxidase, and also oxidative fermentative tests, sugar tests. Okay, so if it's oxidase, then you'll have your Pseudomonas, your Vibrio. Okay, if it's oxidase negative, you proceed with your lactose fermentation, whether it's a lactose fermenter or non-lactose fermenter. If it's lactose fermenter, then it could be your Escherichia coli, Enterobacter, or Citrobacter. If it's a non-lactose fermenter, it could be Serratia, Proteus, okay, and also Salmonella or Shigella. So that was the dichotomous key approach. So you can see here, a mistake in gram staining means the subsequent tests are all pointless because you've already done the mistake in gram staining. So every step is important. Gram staining is important. So even a simple gram staining is crucial in the dichotomous key approach. Okay. So gram staining is what is a step that all microbiologists should be very confident in. So because if you made a mistake here, then you use the different tests for the bacteria, and all the results will be subsequently incorrect. For Blunderbuss approach, the approach is different. It's not sequential. What Blunderbuss approach does is you do all the possible tests, and then you compare the results of all the positive or all your tests against a standard text. Example, a Burgess manual of systematic bacteriology. So instead of doing sequential tests like this, one by one, which also takes time because you need the bacteria to grow, what you do is you do all the tests together. So you do the oxidase, the lactose fermentation, citrate, MRVP, uh, indole. All these tests you do together, and from the results you determine the presumptive identification of your isolate based on standard information that you can get from Burgess Manual of Systematic Bacteriology. So if you remember, the API, the enterotube, the biolog, they use the blunderbuss approach because you are doing all the tests at once, and then you are comparing the results. Is the color chart for the API 20E. So from all the tests that you do at once using the blunderbuss approach, you can roughly compare and determine what bacteria you have. Well, what is the presumptive identification of your bacteria? Of course, if you buy into this system, API20E kits, you'll be given access to their database, and what you do is you just enter the code. All the tests will give you a code, which we will do in more detail during the practical, and then from the code you can determine the bacteria presumptive identification of the bacteria that you have. So this shows the API form. So you have about 20 tests here. Okay. The oxidase test is usually done externally. Okay. So from here to here, these are additional tests. So usually, if you have enough tests until here, is adequate enough for presumptive identification. So if it's positive for OMPG, you tick 1. If it's negative for ADH, you don't it's 0. If it's positive for LDH, you put 4. So the total will be 1 plus 4. It will be 5 here. Okay, so same goes for the other tests. So if it's positive, if it's negative here, and positive here, the code here will be 4. So therefore, from the code, once you have the code here, you enter it into the software. It will give you a presumptive identification. So these are the names of the tests, biochemical tests, and the abbreviation in the API kit.